So the lactose operon, which we've just talked about, is an example of a catabolic operon, where you are breaking down something, and breaking down in this case lactose. An example of an anabolic operon is the tryptophan operon. And this is where we will make something, and what we're making here is the amino acid tryptophan. Now tryptophan is an amino acid that is necessary for growth. And so if you don't have tryptophan, you should make it from some other product. And this operon is normally on. And so we're normally making tryptophan. So here is the operon down the bottom here in this cartoon. Here is the promoter region. This region here. Here is the operator region. The promoter, of course, will bind RNA polymerase as normal. The operator region will bind a repressor protein, a different repressor protein in this case, but it will bind a repressor protein to that area to prevent RNA polymerase from working. And there are 5 structural genes in this particular operon. There's E, D, C, B, and A. And together, the products of these 5 genes create this amino acid, tryptophan. So we want to turn this on if there is no tryptophan in the cell, because then we would want to make tryptophan. And we want to turn this off if tryptophan is present in the cell, because why would you want to turn it on if you've already got it there, right? And so E. coli then makes tryptophan if the cell needs tryptophan. So let's look at the tryptophan repressor molecule. And this is a protein that binds to the operator region. So in this diagram we see some DNA, double-stranded DNA, and this is going to be the operator region of that DNA strand. And here is a cartoon of this tryptophan repressor protein. And if you look at these 2 different shapes, this shape over here where it is not bound to DNA, it sort of looks like this. Its 2 arms, up and bottom arms, are bent in. Whereas if you look at this same protein, when it's bound to the DNA, we see that those 2 arms are quite straight there, and they're binding to the major groove of the DNA double helix. So what has caused the change in shape of this protein such that it can bind to this double helix? Well, if you look at these 2 yellow pizza type molecules here, you'll find that they are stuck in the corners, and what they are molecules of tryptophan. And so they bind in the corners, change the shape of the protein, and that protein is then allowed to bind the DNA. So this is a good example of change the shape and you change the function. The tryptophan repressor then binds to the operator region and stops RNA polymerase from transcribing. So it's a simple on-off switch. But the reason we're looking at the tryptophan operon is that it has a secondary mechanism by which it can control the expression of this gene. This gene also contains a region called the leader region, and it's just between the operator and the first structural gene. It's not very long, it's just in here, but it's called the leader region, and it contains some very special sequences. This leader region, and here's the sequence all throughout here, it starts at the top right and follows the numbers around to here. This is the first structural gene, the TRPE region, and the rest of the gene is off to the right. This leader region contains a special subregion called an attenuator region. Attenuation just means it's a bit like a volume control. You can turn things up and turn things down. So this region here changes the level of expression of all these genes downstream, the EDCBA genes. So this attenuator region has in it this particular sequence. And here's the RNA sequence across the bottom, and that translates into an amino acid sequence across the top here. And as you can see, there's a start sequence, a start codon, and there's a stop codon here. What's special about this particular protein is that it contains 2 codons, UGG, UGG, that require the tryptophan amino acid. Now remember, this gene makes tryptophan. But here we have a leader sequence that actually requires tryptophan. So this requires the product of the gene it is controlling, and this is a mechanism of control and feedback here. Attenuation, otherwise known as fine-tuning. And this leader sequence is 140 base pairs in length, and is actually divided into 4 sections we call 1, 2, 3, and 4. The leader peptide that we just saw in the previous slide, which is 14 amino acids long, covers sections 1 and 2. And how this works is that here are sections 1, 2, 3, and 4, and the leader peptide is right in here. We can see here there's some folding of RNA, and this is to show that a single strand of RNA that we have here has the ability to fold up. And in this particular case, sections 1 and 2 can fold up, 3 and 4 can fold up, and 2 and 3 can fold up. So it might be a little bit confusing, but here's a diagram to help you out. This red line is the messenger RNA of the leader sequence. So what has happened so far is that the repressor protein is not present on the operator. RNA polymerase has transcribed, and it has currently gone through this first region and has made messenger RNA. So the RNA polymerase is currently somewhere near the E gene, the TRPE gene, and this is what's come out of the RNA polymerase. And you can see now it's starting to fold up. Sections 3 and 4 can fold up, and sections 2 and 3 have the possibility of folding up, and sections 1 and 2 could fold up, except that there's this in the way. This is a ribosome, and ribosomes, as you know, translate messenger RNA into proteins. So what this ribosome is doing is translating that small 14 amino acid leader peptide. You see it's partly over section 1 and partly over section 2. So even though 3 and 4 can fold up, 2 and 3 cannot fold up. They're not going to fold up because section 2 is being flattened out by this great big ribosome. And so we can only have sections 3 and 4 fold up. 2 and 3 will not fold. This happens when there is high tryptophan in the cell already. And what's happening, of course, is that this ribosome is reaching those 2 tryptophan codons, and it's translating through there and covering the message. So if we go back to the previous slide, here are the codons for those 2 tryptophans. The ribosome has translated all the way down. It has gone through these tryptophans, added the 2 tryptophans, and continued on, and we have ribosomes that are covering all this sequence. Remember, it's not just one, it's multiple ribosomes. We call them polysomes. And so ribosomes are covering this whole leader sequence. This leader sequence is in sections 1 and 2, and it's being flattened out by ribosomes. So we only get sections 3 and 4 folding up under high tryptophan levels. Let's look at the situation if we have low tryptophan levels. If there's not enough tryptophan in the cell, then when the ribosome reaches that UGG in the A site of the ribosome, it's going to stop, or it's going to stall, as it says here. And when it stalls, that allows sections 2 and 3 to fold up, because the ribosome has stopped right here. If sections 2 and 3 fold up, then 3 is no longer available to fold up with 4, and therefore we have no 3 and 4 folding. Now, why is this important? It's because 3 and 4, this loop here, this is known as a row independent terminator. And if you remember your transcription, then one way that you can stop RNA polymerase from transcribing is to create a row independent terminator sequence, and that is where you have an inverted repeat which folds up on itself to cause a stem loop that folds over and stops RNA polymerase. It terminates transcription. Now, if you have low levels of tryptophan and that ribosome does not make it all the way through section 2, so sections 2 and 3 fold up, you do not get that 3-4 stem loop, because 2 and 3 is not a terminator loop, only 3 and 4 is. And so with low tryptophan, you get no 3-4 folding, which means RNA polymerase continues and you get tryptophan made. So this concept of being able to control the synthesis of an amino acid by having a particular sequence in the leader peptide is present here in the tryptophan operon. But exactly the same system is present in other amino acid operons as well. For example, here is the phenylalanine operon leader sequence. The same kind of system. It has sections 1, 2, 3, and 4 that can fold up. But if you look here, we actually have 7 separate phenylalanine codons, which means that the ribosome, of course, if there's low phenylalanine, the ribosome will stop here, which will mean that there's no termination of transcription, and you go ahead and you make the genes to make phenylalanine. The same thing with histidine. Here are the histidine codons. Again, there's 7 of those, and which would mean that the ribosome would stop right here, and you again would stall out. And so this mechanism that we've learnt about the tryptophan operon, you can also apply the same system to any other amino acid operon present in E. coli.